In our final episode of Neural Frontiers, we are exploring emerging therapeutic modalities that could revolutionize how we treat brain disorders from gene therapy to digital therapeutics. Gene therapy has shown remarkable success in rare neurological disorders, but scaling to common brain diseases presents unique challenges. The brain's post-mitotic neurons can be easily replaced if gene editing goes wrong. So, how do we balance the transformative potential of genetic interventions with the irreversible nature of brain modification? Are we ready for widespread clinical applications?
Yeah, gene therapy has truly transformed the treatment possibilities, especially for rare neurological diseases where correcting a faulty gene can be life changing. But scaling this to common brain disorders presents unique challenges. The brain's neurons doesn't regenerate, so any off-target effects or unintended gene edits could be irreversible, and we must prioritize safety through precision targeting, robust delivery systems to cross the blood brain barrier, and reversible or controllable editing tools as well. While clinical trials are advancing cautiously, widespread application will require not just technical breakthroughs but ethical and regulatory frameworks that prioritize patient protection.
Right. So, cell therapy and tissue engineering offer the possibility of replacing damaged brain circuits, but the brain's exquisite wiring patterns evolved over millions of years. How realistic is the goal of integrating replacement cells into existing neural networks? Are we attempting to recreate something that might be fundamentally reproducible?
Honestly, replacing damaged brain circuits with new cells is an incredible goal, but also tremendously complex. The brain's wiring evolved over millions of years and involves highly specific connections that are difficult to replicate. Some progress in stem cell and others in tissue engineering shows promise for partial circuit repair, but for full integration into existing neural networks remains a major hurdle. We might not recreate the exact original wiring, but instead we can aim to restore essential functions or support brain plasticity, helping the brain to rewire itself with the new cells.
Digital therapeutics, therapeutic app and software can potentially treat neurological and psychiatric conditions without traditional pharmacological interventions. Yet, regulatory pathways for software based therapeutics remain unclear. How do we validate and regulate therapies that exist purely as code? And should digital interventions be held to pharmaceutical standards of evidence?
Digital therapeutics represent a whole new class of treatments that don't fit traditional drug paradigms. Regulatory pathways are evolving, but not fully redefined for software as medical devices. Validation requires rigorous clinical trials demonstrating efficacy and safety, transparent algorithms, and cybersecurity protections also. While some argue digital apps should meet pharmaceutical evidence standards to protect patients, there is also a need for innovation friendly framework that allow iterative improvement and integration with real world data. Collaboration between regulators, developers, and clinicians is crucial to create balanced and clear guidelines.
Right. So, psychedelic compounds are experiencing a renaissance in neuroscience with promising results in depression, PTSD, and addiction. However, these drugs work through fundamentally different mechanisms than traditional pharmaceuticals, often requiring therapeutic context and integration. How do we develop psychedelic medicines within existing pharmaceutical framework? Are we forcing square pegs into round holes?
Psychedelics challenge traditional pharmaceutical models because their effects rely on complex therapeutic contexts and psychological interactions. These drugs aren't just molecules acting on receptors. Their benefit often come from how they are administered and the patient's mindset also. Developing them within existing frameworks means considering both drug and psychotherapy together, which is hard to separate in clinical trials. Regulatory agencies are beginning to adapt, creating hybrid approval pathways that recognize these nuances while ensuring safety and reproducibility.
So, brain organoids and organs on chip offer unprecedented opportunities to model human brain disorders in vivo. These systems could revolutionize drug screening and reducing animal testing. However, they lack the full complexity of living brain. How sophisticated must these models become before we can trust them for drug discovery decisions?
Brain organoids and organ on chips have opened unprecedented windows into human brain biology and disease modeling. Though they lack full brain complexity, such as full vascularization and maturity circuitry, they provide scalable human driven platforms for early drug screening and mechanistic studies. The goal isn't to replace animal models entirely, but to complement them, reducing animal use and improving prediction of human responses. As these models become more complex and validated, trust in their predictive power should grow, especially when integrated with other biological and the computational data.
Right. So, neuromodulation technologies from deep brain stimulation to transcranial stimulation offer non pharmacological approaches to treating brain disorders. As these devices become more sophisticated and less invasive, could they replace traditional drugs for neurological conditions? How do we integrate device based and pharmacological approaches?
Neuromodulation technologies are advancing rapidly, providing non pharmacological ways to treat conditions like Parkinson's, depression, and epilepsy. As these devices become less invasive and more sophisticated, they could indeed reduce or replace some drug treatments. But many brain disorders will likely require a combination approach: devices to modulate circuits, drugs to target molecular pathways, and behavioral therapies to optimize functions. Integrating these modalities require interdisciplinary research and clinical strategies that personalize treatment to each patient's unique brain dynamics.
Jhansi, this journey through neuroscience drug discovery has revealed both immense challenges and extraordinary opportunities. To our listeners in academia and industry, the future of brain health depends on bold thinking, innovative approaches, and sustained commitment to solving the most complex puzzles in medicine. Thank you for joining Neural Frontiers.
Thank you, Ritika, for this thought provoking session. It's clear that neuroscience drug discovery is a complex but promising field. I appreciate your insights and perspectives on the challenges and opportunities ahead. To everyone listening, I encourage you to stay cautious, collaborate, and push the boundaries of what's possible in neuroscience. The future of brain health is indeed bright, and I'm excited to see what advancements will come from the collective efforts of researchers, clinicians, and industry professionals.
Thank you.
Thank you.